Welcome back, Biomonsters. Today we're going to be talking about the cell cycle. Specifically, we're going to be talking about what allows cells to divide so they can aid in the process of growth, repair, and also sexual as well as asexual reproduction. All right, here we go. The first thing that we're going to do is have a quick nucleus review. Let's see how much you'll remember about the nucleus as an organelle. So let's look at that first question. It says, what important information is stored in the nucleus? Well, the important information that's stored in the nucleus, and the reason why it's considered the control center of the cell, is because it contains DNA. That's the important stuff that's housed in the nucleus. What can you say DNA is like? Well, we've talked about this before. DNA is a double helix. But with all that DNA inside of the cell, it's also like a big pile of yarn or string. The next thing that we need to ask ourselves is, what does DNA do? Well, DNA has one job. It contains hereditary information. That means it controls every process that takes place inside of a living thing. And then lastly, the question is, how is DNA packaged? Well, DNA is packaged, if you remember, in specialized structures that kind of look like butterflies or X's. And these structures are called chromosomes. And we're going to spend some more time talking about chromosomes and the substructures that make up chromosomes as we move further along in today's discussion. All right, now that we've had a little bit of a review from what the nucleus contains as well as what DNA does and what it essentially looks like, we want to talk about some new information. So here's some new information. So before a cell copies itself, you need to know what the DNA looks like. And we talked about it before. We said the DNA is like a big pile of yarn. So basically it looks like this. Now the problem with this is that if the cell needs to divide, it has to be able to precisely or exactly separate the DNA so we know exactly how much DNA is going to one cell and how much is going to the other. So what's the big deal, or what's the problem with having a big pile of yarn? It's hard to separate, right? And we need to be able to precisely or exactly separate our DNA as we are making new cells, because we want those cells to be genetically identical to one another. Well, the great news is that the body is actually found a way, and the cells have found a way to precisely separate DNA. And the way that we're going to do that is we're actually going to manipulate the shape of DNA or change how it's organized. So to make DNA easier to copy and divide, a cell was going to coil or fold. So this word coil, we could also use the word fold, up all of its DNA into structures that have a specific name. This new structure that we're going to talk about is what we just talked about in the review, is called a chromosome. So here's our picture. Let's go ahead and label this so that we have our analogy to help our brains understand this information. This over here is our loose DNA, and this loose DNA equals a pile of yarn. Well, if I had to separate this pile of yarn, it would be pretty difficult to make sure that the new cells would actually have the exact same stuff inside of them. So instead of just having this pile of yarn, what I'm going to do is I'm going to take this pile of yarn and I'm going to organize it into a new shape. So I am going to use chromosomes to organize my DNA. Or instead of having a pile of yarn, I'm going to turn those pieces of yarn into socks. So I can clearly look at this sock and say that this sock is the same shape, size, and color as another sock. Or I could easily say that it's not the same size, shape, and color as another sock. So I'll know whether or not I have the exact same DNA or the exact same genetic information going into different locations. So keep that analogy in mind, because that's the analogy that we're going to work with today as we talk about the cell cycle. So now that we've talked about how cells are, or how DNA is packaged inside of a cell, we need to talk about the actual parts of a chromosome. And so what you need to know is that chromosomes are made up of DNA, which we talked about before. Specifically, it's made up of DNA and proteins. And these proteins are going to help it change its shape and fold into the specific conformation. Each chromosome is also made up of identical parts or identical sides that are called chromatids. So if I'm going to draw my chromosome, and you guys should be drawing the same thing on your paper. So I'm going to draw my chromosome here. When I'm talking about my two sides, I'm going to draw an imaginary line right down here so that I know this side is exactly like this side. And because these two sides are identical, we're going to call these our chromatids. So this is a chromatid, and this is also a chromatid. And because they're identical, we can actually add a word here. We can say that they are sisters. So sister chromatids. The last thing that we need to talk about is how are these two chromatids held together? So you'll notice they're held together in the middle here. And this actually has a specific name. It's called a centromere. And the great thing about this term is that centromere sounds like the word center. So if you even wanted to say centromere to help you remember, that would be great too. So I'm going to go ahead and label this as my centromere. And again, remember this whole thing is called a chromosome. So now that I know the parts of my chromosome, all right, so now let's talk about some specifics. So human body, also known as somatic cells, have a magic number, and their magic number of chromosomes happens to be 46. So human body cells or somatic cells have 46 chromosomes in them. So we can say that's like 46 socks in keeping with our analogy, right? These socks over here. Now, these socks or these 46 chromosomes can be put into pairs, and in fact, they are put into pairs. So we can say, instead of just saying that we have 46 chromosomes, we could also say that we have 23 pairs. Or instead of saying that we have 46 socks, we can say that we have 23 pairs of socks. Now these pairs have a specific name. So each pair of chromosomes, and this is important to know, are roughly the same size, shape, and they have the same function. Because they're roughly the same size, shape, and have the same function, we can call these guys homologous chromosomes. So even though we have 46 chromosomes, we have 23 homologous chromosome pairs. And now the next thing that we need you to understand is that you get each half of the pair from two different individuals in sexual reproduction. So for you, humans, each pair you get one chromosome from your mom, and the other chromosome pair you get from your dad. So let me quickly draw you what this might look like. So here's one chromosome. And this chromosome has a twin that's roughly the same size, shape, and controls the same function. And because they're roughly the same size, shape, and control the same function, we can say that these two chromosomes are homologous chromosomes or homologous pairs. And we know that one came from mom and the other one came from dad. Now let's quickly review our analogies just so that nobody gets confused. Remember that we're starting out in our cells with a big pile of DNA that's loose and tangled like a big pile of yarn. To make it easier for us to separate, to prepare for cellular division, what we're going to do is we're going to take that loosey-goosey DNA and actually put it into a structure that's easy to identify. So we're going to put it into socks or chromosomes. And we're going to have 46 of them because that's the magic number for humans. But all 46 chromosomes have a matching pair that's roughly the same size, shape, and controls the same function. And we call those homologous pairs or homologous chromosomes. So we can take our number 46, which is the magic number, and say that we have 23 pairs of homologous chromosomes, or we can say we have 23 pairs of socks. All right, now go back and look at your notes. See if you can answer the stop and jot questions on your paper. When you think you have the right answer, raise your hand and your teacher will come around to check for correctness. Next thing that we're going to talk about are what are the different types of chromosomes, because there are different types of chromosomes that control different things in the body. The two types of chromosomes that you're responsible for knowing in our class are chromosomes called autosomes, as well as chromosomes called sex chromosomes. And we're going to talk about each one of these types of chromosomes separately. So let's talk about autosomes first. So the first 22 pairs of chromosomes are referred to as autosomes. So autosomes are chromosome pair 1, 2, 3, 4, all the way up to pair 22. The unique thing about autosomes is that they can control lots of things: hair color, eye color, the shape of your nose, how tall you're going to be. But there's one thing that autosomes never control. So autosomes do not determine gender. So they have nothing to do with whether you're going to be a male or a female. Now that's not the case for our sex chromosomes. So the 23rd pair, or we can say the last pair of chromosomes, are called our sex chromosomes. And sex chromosomes can look one of two ways depending on whether you're male or female, because sex chromosomes determine the gender or sex of the individual. And you guys need to know what the sex chromosome pairs look like for females, and you also need to know what they look like for males as well. Now for females, the sex chromosome pair will be XX, and for males, it will be XY. Now the reason why this is the case is because mom gives an X, and dad also gave an X to make a female. The unique thing is that mom can only give X's. So the male actually determines the sex of the child. So the dad is in charge of that. So again, if you're a male, mom gave you an X because it's the only thing that she could give you. But dad, instead of giving you an X, gave you a Y. So dad gave you a Y here. An easy mnemonic device to remember that men are always XY, so that you'll know what your sex chromosome pair should be, is to think guy Y. So guy Y, so you'll never forget your sex chromosome pairs for females and also for males. All right, when you come to another stop and jot, please look at your notes, answer the questions on your day sheet. When you think you have the right answer, raise your hand and wait for your teacher to come around and check your answer for correctness. All right, our next topic that we're going to talk about are karyotypes. Now, this is a really weird word, so let's talk about it. Each organism, all living organisms, have a characteristic number of chromosomes that can be identified by something called a karyotype. And a karyotype is really just a fancy picture of chromosomes. Now, if you recall from earlier in our video, we told you that the magic number for humans is 46. So if you were to look at a normal, genetically normal human being's karyotype, which is just a picture of all of their chromosomes, you would expect to see 46 chromosomes in that picture. So a karyotype is just a picture of all the chromosomes within a cell by pairs in a fixed order. So let me show you a specific example. If you look at this picture here, there are a bunch of chromosomes. Now these chromosomes look a little bit different than the chromosome that I showed you before because the other side is missing. Well, what we can do with this picture, so we can take it and we can organize it. And when we organize this picture, we'll put all of the first pair together, the second pair, the third pair. And remember that these two pairs are roughly the same size, shape, and control the same function. So this is a homologous pair, right? Or homologous chromosomes. And we have a total of 23 homologous pairs, or a total of 46 chromosomes. And they're lined up based on size. So the first 22 pairs are going to be our autosomes, and then our last pair are going to be our sex chromosomes, like we talked about before. So a karyotype is interesting because a karyotype can tell us one of two things. They can tell us whether or not the karyotype comes from a male or a female by looking at the sex chromosomes, and it can also tell us whether or not the individual is genetically normal because we can see whether or not they have the magic number or 46 chromosomes. So let's go ahead, take a peek at the picture, see if you can figure out whether this is a male or a female, and also see if you can identify whether or not that this is a genetically normal karyotype if there's actually 46 chromosomes. So now that you've had a chance to look at that, you should have figured out that there are 46 chromosomes in this picture, or 23 pairs. So far it looks like the individual is genetically normal. And if we look at the last set of chromosomes, looking at our sex chromosomes, you'll notice that we're XX. So this means that this is a karyotype of a human female. Let's practice with another. So again, we have a picture of a bunch of chromosomes that are all jumbled. We need to lay them out in an organized fashion. We're going to put our autosomes as our first 22 pairs, and then our last pair is going to be our sex chromosomes. Again, you're looking to count to see how many we have, and you're also trying to determine whether it's a male or female. So if you've counted correctly, you should have noted that there are 46 chromosomes or 23 pairs, so that means that we're genetically normal. And if we look at our last pair of chromosomes, you notice that we have an XY, guy Y, so that means that this is a karyotype of a human male. All right, here's your next stop and jot. Go ahead and pause the video, see if you can answer the questions on your day sheet. And when you think you have the right answer, call your teacher over to check for correctness. Quick review before we move on. The first thing that I want to remind you of is the key component of the cell theory, that all living things are composed of at least one cell, which we've talked about really since the very beginning of the year. Now, if all living things are composed of at least one cell, that means all living things have to be able to divide to make more copies of themselves in order to ensure that three things can happen for that individual or for that organism. The first reason why we have to have cells, their cells have to be able to divide, is to be able to repair damaged cells. So cells get damaged all the time. If you get a paper cut, those are damaged cells and they have to be replaced. If you've ever had a burn, skin is constantly being sloughed off and has to be repaired, right? So we have to be able to repair. The next thing that we have to be able to do is we have to be able to grow. And we can't grow unless our cells are dividing. So think about you. Many of you, when you were born, were somewhere between 6 pounds and maybe 12 pounds at the max, right? And you guys are clearly much heavier and much larger than you were when you were born on your original birth date. The only way for you to actually get bigger, because we're not growing the individual cell size because that wouldn't be efficient. Think about our Walmarts versus our Reed's grocery stores. The only way to actually grow the organism is to make more copies of the cells. So growth is also another reason why we do cell division. The last reason why we do cellular division, and this is not true for us, but it's true for other types of organisms, is for something called asexual reproduction. And if you look at this picture here, you'll notice that we have a single-celled organism. This happens to be an amoeba. This single-celled organism is simply dividing in half and making an identical copy of itself. So all of these processes, regardless of why we're doing it, we're making identical cell copies. That's really important. We want to make the exact same thing that we started with. So asexual reproduction actually makes things called clones. So there isn't a whole lot of genetic diversity, and in fact, we don't want any genetic diversity from one generation to the next when we're talking about cellular division in this regard. All right, so now that we've talked about the cell cycle or cellular division in general, let's talk about some specifics. So the cell cycle is a time for growth and division of the cell. So you need to know what the cell cycle is actually composed of. So the cell cycle allows us to make exact copies, we talked about this before, of the starting cell. Basically, it's a fancy copy machine or Xerox machine. Now the cell cycle is divided into 3 periods. The first period is interphase, and if you look at our cell cycle here, it's this part here. It's the largest part. So interphase is the longest part of the cell cycle, and most cells spend most of their time in interphase. Mitosis is the second period of the cell cycle, which actually allows for cellular division or division of the nucleus. And then finally we have cytokinesis, which is the last period, which allows us to actually divide the cytoplasm so we can end up with 2 new identical cells. So let's go ahead and dive in and talk about each one of these periods separately. The first period that we're going to talk about is interphase. And the most important thing that you need to know about interphase is that it is the longest period. So if you had to guess what phase of the cell cycle a cell was in, it would be a pretty good guess to say interphase, because that's a majority of the time where a cell spends its time in the cell cycle. Here is where cell growth happens, metabolism, so normal chemical reactions are taking place. We're copying DNA in preparation for cellular division, and we're also copying organelles, things like mitochondria, ribosomes, the Golgi apparatus, and endoplasmic reticulum. I'm going to quickly draw a picture of what interphase would look like. This is a simple text to show you, and you guys should be copying this down on your notes as well. So in interphase, we're going to have our nucleus, we're going to have our cell, and our DNA is not going to look fancy. It's going to be loosey-goosey, just like this. So it's going to be a big pile of yarn. So this is our DNA. So it's our loose DNA. So anytime you're seeing loose DNA, you should say to yourself, well, we're definitely in interphase, because the only time that we're going to package our DNA up into our chromosome structures is when we're preparing to divide. All right, now that we've talked about interphase, let's move on to the second phase, which is mitosis. And mitosis has 4 subphases that you need to know. These 4 subphases are listed right here in order, and you do need to know them in order. The easiest way to remember the order is to think about the first letter of each one of those phases and come up with a mnemonic device or a memory clue. And what we like to say is PMAT. That's the easiest way to remember it. So we have prophase, metaphase, anaphase, and telophase. And we're going to talk about each one of those phases, and it's your job to know what's happening there and also to be able to identify what phase of mitosis a cell is actually in. All right, so let's talk about prophase first. So in prophase, some key things that are happening here are chromosomes are becoming visible. The nuclear envelope, or the outer portion of the nucleus, starts to dissolve and disappears. And we're also going to start to see spindles form. So let me show you what this actually looks like. And again, you need to be drawing the same picture in your notes. So here's my cell membrane, or the outer portion of my cell. And on my previous picture in interphase, you'll notice that I drew a clearly defined nucleus with lots of loosey-goosey DNA in the middle. That's not what we're going to do here in prophase, because we know that our nuclear envelope is starting to dissolve. And because our nuclear envelope is starting to dissolve, I'm going to draw it like this. So that's my nuclear envelope starting to dissolve. Another key characteristic is that we're actually going to see chromosomes now. So instead of our DNA being loosey-goosey, we're going to see our DNA in the shape of chromosomes, just kind of randomly floating inside of our nucleus that's starting to dissolve. We also know that we're going to start to see spindle fibers form. And so our spindles are going to be these guys that are starting to form along the outside of the cell. Our next phase is metaphase. And metaphase is a really easy phase to identify, because in metaphase, we're thinking about the word M. Metaphase, the chromosomes line up in the middle, or the equator. So whenever you think about metaphase, we want you to think middle. Metaphase, middle. Metaphase, middle. So here's what it's going to look like in our drawing. And again, you need to put the same drawing on your paper. So at this point, my nuclear envelope has completely disappeared, but my chromosomes are going to start to do this interesting dance, and they're going to arrange themselves along the middle, or the equator, of the cell. So here are 4 chromosomes. They're arranged along the middle, and we have our spindle fibers now that are starting to attach to our chromosomes, specifically to the center, or the centromere, of our chromosome. So again, metaphase, middle. That's the easiest thing to remember. Metaphase, middle. All of our chromosomes are lined up right down the middle of our cell. Now in anaphase, we're going to start to see something unique as well. Chromosomes are going to start to split, so we're going to break our chromosomes apart, and the 2 chromatids are going to move in opposite directions. In order to do that, our spindle fibers actually have to shorten, and when they shorten, they're going to start to pull my chromatids away from one another. So let's draw a quick picture so that we can see what it actually looks like. And again, you need to be drawing the same thing in your notes. So initially in our last picture, all of our chromosomes were lined up in the middle. But here, our chromosomes are starting to be pulled apart from one another. So we still have the same number of chromosomes, but now they've broken apart. And the reason why they've broken apart is because they're being pulled to opposite ends of the cell by our spindle fibers, which are these guys that are attached to the center, or to the centromere. Now, to help us remember what's actually happening here, I'm going to go ahead and draw in an arrow. These chromatids are moving in this direction, and the chromatids that were on the left-hand side are going to start to move to the left-hand side of the cell as well. Remember, the whole point of doing this is to separate our genetic material. Our last phase of mitosis is telophase. Telophase is basically just the opposite of prophase. So if you think about prophase, the nuclear envelope started to disappear. So in telophase, we're going to start to see them reappear. Chromosomes turn in or coil up and become condensed in prophase, and here they're going to start to relax and loosen back up again into loosey-goosey DNA. Instead of making our spindles, we're going to start to dissolve them, break them apart because we no longer need them, and the cytoplasm is going to start to pinch in. So let's draw a picture and see what that actually looks like. So here is my cell. Now I'm going to start to have my nuclear envelope start to reform. I'm going to have my loosey-goosey DNA again. It's no longer chromosomes because I don't need to worry about separating it. And what's going to happen is here we're going to start to see the cell pinch in a little bit and pinch in a little bit right here. Our cell membrane is going to start to pinch in because we need to start, now that we've separated the nucleus, we need to start thinking about separating the cytoplasm, which brings us to our last phase of the cell cycle, which is cytokinesis. And if we look at this term cytokinesis, cyto sounds like the word cytoplasm. So cytokinesis is basically the division of the cytoplasm after we've already separated our genetic material. So if this were actually happening in a cell, it would look something like this. So we have our nucleus here with our loosey-goosey DNA, we have another nucleus here with our loosey-goosey DNA, and the cytoplasm is pinching in here until eventually we're going to have two separate cells. And when we have these two separate cells, they're going to be genetically identical to one another. So here is a quick animation showing you what we've just talked about. So in this video, the very first thing that's going to happen is prophase. So our DNA is going to start to coil up into something called chromosomes. Our spindles are starting to form. Our nuclear envelope has disappeared. Now we're going into metaphase, and in metaphase we're going to line up our chromosomes along the equator or the middle of the cell. Anaphase is going to happen where we're splitting our chromosomes apart, so ana split, kind of like banana split. And then lastly, we have telophase, and in telophase, we're just undoing what happened in prophase. So our DNA is becoming loose again. We're going to reform our nuclear envelope. And then the last thing that's going to happen is cytokinesis, where we actually pinched off and separated our cytoplasm from one another. So at the end of this process, we end up with two identical cells from the original one that we had at the beginning when we started mitosis. All right, guys, we're at your last stop and jot. Go ahead, look at your notes, see if you can answer the questions on your day sheet. Raise your hand when you think you have the right answer so that your teacher can come over and check for correctness. Thanks so much, and make sure you study your notes. Good luck.